Okay, so hi again. As I was saying, today we are going to be going through some basic information about the course. And then we're going to do the study guide and take up some questions, and we'll do some practice problems for the quiz and tests as well. If you have any question at all during the session, you can feel free to unmute yourself. Or you can just type the answer, type the questions in the chat box. Okay, so hopefully you have had a chance to look at the course webpage, and everything you need for the entire course is already posted on the webpage. Okay, so there is a recommended schedule, right? That you do lecture 1 this week, lecture 2 next week. However, if you feel like you are ready to move on to learn ahead, all the materials are posted. You are welcome to do that. Okay, so let's take a look at what you have to do for each class. The first thing you should do is to download the weekly schedule. So there is a quick link button here that you can click on, and it will take you to the weekly schedule. Okay, so here is what it looks like, and it will tell you when everything is going on in the class. So for example, you can see we are here today, right, doing the synchronous tutorial, and the time block is 2 hours. Okay, and on your timetable, you will also see that you have a class, the same class on Tuesday, also from 8:30 to 10:30. So that is for asynchronous learning. So as I mentioned in the email, that's when you are going to be watching the lecture recording that I have posted. Okay, so I just want to do a quick poll and see if you guys have had a chance to watch the lecture recording. If you have watched the lecture recording, please select A. If you have not, please select B. Sometimes it looks like I'm looking the other way. That's because I have 2 screens. It's not that I'm distracted or anything. I'm just checking to see if people are posting anything in the chat room or that sort of stuff. Okay, so that's good. A lot of you have actually watched the recording, and that's great because the recordings are kind of like the full thing, right? Kind of like the entire movie, if you would. And what I'm going to be doing in the tutorial, it's more like a recap. Okay, so I will be going. I won't be covering all the materials in the lecture. I'll just be focusing on the study guide, and sometimes. I won't be able to go into as much detail as I did in the lecture recordings either. So you have to watch the recordings before coming. And if sometimes you aren't able to do that, come to the tutorial anyways, because it is beneficial for you. Okay. And like I said at the beginning, the entire tutorial is going to be recorded, and I'll post it online, and you can access it after. So almost all of our synchronous sessions happens on the Friday, except for, let's see if there are any exceptions. Yeah, except for like when it's before a test. Okay, so your first test is not until October 29th. And then for that week, our synchronous session will be on the Tuesday, because I want to do a test review with you before you write the test. Okay. But I will remind you, okay? I will communicate with you primarily through emails, and you should check eCentennial on a regular basis, because I'll be posting an announcement, all right? So usually I post an announcement at the beginning of the week as well as after the tutorial, to kind of tell you, what you have to do for that particular week. But generally speaking, right, like you would watch the lecture recording, the corresponding lecture, and then you come to the tutorial. That's basically how it is. Okay. So in terms of the where to find everything, I can quickly show you. If you go to the course homepage, let me switch to student view, so I see what you see. So this is what you will see, and here are a bunch of quick links that you can click on. But the best way to find all the material is if you click on content, and then lecture, and here would be the folders for all the lectures. Okay. So in the lecture 1, scientific method, you would see the lecture recording folders, So there's the PowerPoint that you should download and probably print it out if you could. And then you would take notes while you watch the lecture recordings, okay? Now, sometimes the recordings will also require you to download the study guide, okay? In some of the later units, when we do like mutations and stuff like that, there are questions that I would be doing in the lecture recording that requires you to have the study guide, okay? And this is the study guide that you should print if you have a printer to print and bring it to the tutorial, because that's what we will be filling in, okay? You could try to do it on your own first, but if you're busy, you can wait until the tutorial session and we'll do it together, okay? There are additional resources. There are some crosswords you can do to help you remember the vocabularies. There are some self-quiz that you can do as many times as you like, just as kind of like a formative assessment, right? To see if you've learned the material, okay? And then there are these video resources. So if you open the video resources, it'll take some time to load, but it will take you to a collection of videos that I found online, and it relates to the particular topic that we're doing, okay? So sometimes you would find it helpful to look at some of these because it explains things from a different perspective, okay? And if you want to comment on any of the videos, you can type the comment here, okay? So and then I will be moderating this is called Padlet, okay? Padlet. So I'll be moderating the Padlet, okay? But basically, it's a collection of resources for you to look at. So going back to the folder, let's see what else is there. There's also a supplementary reading that you might want to look at. If you click on that, it will open up to an online. You have to click open a new window as well. That will take you to an online open source textbook, and we are not gonna. We don't have a physical textbook that you have to purchase. Okay, if you have. If you want to read about the stuff that we are learning, you can just use this online textbook. And I will only be testing you on the lecture material, so the PowerPoints, the lecture recordings, as well as what I cover in the tutorial. So if there's any additional information that you read about on the online textbook that we didn't cover, or if you see something in one of those videos that I post in the Padlet, you will not be responsible for them. For the quiz or for the test. Do you guys have any questions so far before I continue?
Sorry, can you show me how to get to those videos again?
Absolutely. So you would go to the content and then choose the lecture that you want, say lecture 1, and then study resources. And then there's the video resource. Okay. So there's a video resource for each of the lecture. Lecture 2 would have its own collection and so on and so forth. All right, hope that helps.
Thank you.
Yep. And let's talk about the labs, okay, as well as the breakdown of the course. The course is going to have basically 2 parts, okay, the first half and then the second half. So for the first half, we will have 2 quizzes. So quiz 1 and then quiz 2. Each of them will be 10%, okay? And quiz 1 will be on lecture 1 and lecture 2. Quiz 2 will be on lecture 3 and lecture 4. And then we will have a lab, okay? So the first lab that you will do is the beetroot lab. Beetroot lab. These are virtual labs, so you do not have to come to campus. I'll show you where the labs are in a bit. And that is also worth 10%. And then you will have a test. Okay, test number 1. And test 1 is 20%, which would be lecture 1 all the way to lecture 5 plus the lab. Okay, lab. So that is the first half of the course. And then after the test, you can forget all about it. Okay. Because the second half is going to be non-cumulative. So quiz number 3, also 10%, is going to be lecture 6 and lecture 7. quiz number 4 is going to be another 10%. Lecture 8 to lecture 9. You don't have to copy this. It's actually all in the calendar. And then there is the microscopy lab, which is another 10%. And then finally, test 2 will be lecture 6 to lecture 10. And it will be 20%, just like before, plus the lab. Okay, so together it's going to be 100%. Okay. As to when the labs will be happening, you can look at the weekly schedule, right? Sorry, I lost everything when I restarted my computer. So just give me a moment to pull up the weekly schedule for you guys. 204 right here. There it is. So the first quiz is going to be on week number 3. Okay, and I will be posting information about the quiz as we get closer to it. I will tell you, how many questions there are, and the time and stuff like that. But generally speaking, a quiz you're gonna have about 40 minutes to do, and a test you're gonna have about 70 minutes to do. If you require accommodation, if you're a registered student with CALS, student centers for people who need accessibility accommodations, then you can email me, contact me, and then I will adjust the time limit for you accordingly. Okay, but typically the quiz or the test will be available from 10 a.m. to 10 p.m. So you log on to eCentennial and you would access the quiz during that time frame. And once you start, the timer cannot be stopped. Okay, so even if your computer crash or something like that, the timer will still be continuing. Even if you close your browser, it will keep on going. So it's a good idea to find a quiet place, make sure that you're not going to be disturbed for the quiz to do it. Now, having said that, things does happen, as you saw this morning with me, right? Like my computer literally never crashed. This is like the first time it crashed since online learning. Okay, and if your computer crashes during the quiz, the test, and you cannot log back on, or if the internet goes down, it's okay, right? Don't panic. Just email me. I will try to accommodate you as best I could. Now, if your computer crashes every time there is a quiz, then I get a little bit suspicious, right? Okay, but if it just happens once, you can let me know and we can work something out. Okay, I'm a pretty laid-back person, so feel free to contact me at any time if you have any concerns at all, and don't be afraid to ask questions. Okay, in terms of what is on the quiz, well, before that, did your other teacher show you how to access the quiz? Should I go through that? I mean, it's a Friday already. Like you guys would have most of your other courses already. Did you guys learn about how to access the quiz on eCentennial in your other courses yet? No? Okay. So usually I will post a link for you to go to the quiz. Okay. When I post the announcement, you see there are all these like blue stuff, right? So these are the things you can click on. They're called hyperlinks, right? So on the quiz, I will set up a hyperlink for you to click on. But if you're curious, you can just click on assessment and then quizzes. Assessments and then quizzes. So here are all the self-quiz that you can do, as many to see unlimited, right? As many times as you like for practice purposes. Those are not for marks. And then here you will see a bunch of quizzes that it says future quizzes, right? Because they are going to happen at a later time. And it will tell you it's going to be available on September 21st, 10 a.m. to 10 p.m. Okay. And then you will have one attempt for them. So if you click on them, it will say the quiz period has not started yet because we're not September 21st yet. And then there is the time limit for it. And then when it's the right time, you just click start and then you just do it. Okay, it's quite simple. In terms of what's on the test or quiz, there will be three types of questions. There is just standard multiple choice questions. So that would be a majority of the test. I cannot give you an exact number because each quiz kind of varies, but I would say like 70% of the questions would be multiple choice, right? So that's your standard type of question. There's going to be what we call multi-select question. Multi-select. so or MS, the multi-select questions are going to have more than one correct answers. So in order to get 4 marks, you would have to choose all the correct ones. Okay, so this would be like 2 marks each, 2 marks each. This would be 1 mark each. And then there are application questions. Application questions. So these questions also worth 2 marks. So these are questions that are not as straightforward, and you would have to, apply what we've done in class in order to answer them. Okay. So these are the questions that kind of adjust for the advantage that students get due to the online environment. It's okay to have your notes open. Like, I don't mind that. Okay, it's understood that it's an online environment. You would have access to your notes. You might even have access to Google. I'm not encouraging it, but I know people, have access to these things. So to kind of, level the playing field a little bit, we include these multi-select questions and application questions. These are not things that you can easily search up on the internet. Okay, these are things that you actually have to, do some studying in order to figure out the answer. So that's like, 30% of the test or quiz. So that's the evaluation. Anybody have any question? No, we are doing good. Fantastic. Okay, let's see what else we have to talk about. I think that's it. I don't think there is anything else to talk about. One more thing here, like the virtual tutorial, you can click on that and it will take you directly to this document. There is a Zoom link, and then there is nothing else right now because we haven't done any tutorial yet, right? So after today, I will post a link for today's recording. And then next week, I'll post another one. So you will have an archive, a collection of tutorial recordings. Because sometimes you want to go back and listen to some of the things that I explain again. Or if you join late or if you miss a session, this is where you can find the recordings. So there's also a FAQ for common questions people want to ask. And I think that's it. We're gonna get started. That's my email, of course. I reply email quite quickly. Some of you already contacted me, and I reply almost right away if I'm around my phone. And if for some reason you email me and you have not heard back from me for more than a day, okay, that means I did not receive your email. Okay, I always reply your email if I receive it. Okay, there are other Vincent Wong in the college, so sometimes students send it to the wrong person for some reason. Okay, if you're gonna email me, you can use this one, or there is the other one that is posted on the eCentennial, the one that says VWong41. That one also works. Okay, but bottom line, if I don't reply you don't hear back from me for more than a day, that means I did not get your email. Okay, and then you will just have to email me again. Okay. Yeah, so that's it. Let's get started with today's lesson. So we will be doing lesson 1, which is scientific method. Some of you might came from high school and you just did your high school biology, and these things will be relatively familiar to you. Some of you might be out of school for some time, and you're just coming back, or you have never taken biology before. Okay, so my job is to treat everybody, all of you, as if you've never learned biology. Okay, so I'm gonna start at a very basic level, and if you've done these things before, then maybe you can help out with answering some of the questions. But I'm gonna assume none of you have done this before. So, does everybody have a copy of the worksheet in front of them? Let me just do another quick poll. Do you have it? If you have it, press yes. Press A. If you don't have it, press B. Okay, most of you have it. For the people who don't have it, do you know where to get it? You can just unmute and tell me. It's faster that way. Do you know where to get it? I have a few people who do not have the sheet.
No.
Okay. So do you know how to go to the eCentennial homepage? This one, right?
Yes.
Yeah.
Yes.
And then in the if you scroll down to the announcement here, this is the link you want to press. Okay, print and attempt to complete this one. So you just click on it and it will take you right there. Okay, so that is where you will get it. All right, let's get started. So these questions are the ones that you should be studying for to prepare for the quiz or for the test. First question, list 7 characteristics of living organisms. Okay, so in the PowerPoint, there were a bunch of characteristics. So the first one that I can remember is that they exhibit order. Okay, so what does exhibit order means? So there are some, some structures to the organism. Structure. There are some maybe patterns. There is some symmetry. Okay, and complexity to it. Okay, so that's what organisms are going to have. Right? If you think of like a person, there's like left-right symmetry. If you think about flowers, there are patterns on them. If you think about like a cow or something, there are the patches, right? These kind of patterns. So all these complexity, pattern, structure, we call them exhibit order. Another one that I could think of is the ability to maintain homeostasis. Can anybody tell me what the word homeostasis means? Anybody? You can unmute if you feel comfortable, or you can type it in the chat box. What is homeostasis? Anybody know what homeostasis means? That's okay. So homeostasis means, yes, so thank you. Someone type in the chat box. Is it a caramel? Is it, am I pronouncing it right? Apologize if I'm making a mistake there. So the ability to maintain internal balance. Okay, so ability to maintain homeostasis. For human beings, we have to make sure that our internal body temperature is 37 degrees, and we have to do various things to make that happen. Now, in terms of homeostasis, there are 2 ways that organisms can maintain homeostasis. They can have physiological behavior, a method, or there is behavioral. Okay, so physiological means the things that your body does automatically to maintain that balance. So let's say you're very cold. You go outside in the middle of the winter and it's really cold. An example of a physiological mechanism to help maintain homeostasis would be shivering. Okay, so shivering creates muscle contractions and that heats you up and warms you up. A behavioral response to maintain homeostasis when it's cold would be you putting on a jacket. Okay. Let's see what's another characteristics of living things: growth and development. Okay, so this one is pretty self-explanatory. We grow, right? We were much smaller when we were babies. Now we are quite big. Next one, requires energy. Okay, so humans, animals, we have to consume food to get energy. Plants, they use the sunlight as the source of energy. They do photosynthesis, for example. Another one is they respond. All these are in the PowerPoint, right? By the way, right? We're just kind of reviewing. Respond to stimuli or stimuli. Plural is stimuli. So that is a change in the environment. Okay, a change in the environment. So sometimes the stimulus is going to prompt a behavioral or physiological mechanism to maintain homeostasis. So If the stimulus is that it is cold outside, then you're going to shiver or you're going to put on a jacket. Okay. Another one is reproduction. Reproduction. Organisms are able to reproduce their own kind, which means like humans make baby humans, and then pandas make baby pandas. Bacteria make clones of themselves. Okay? If it's a living organism, they are going to be able to replicate and create more of the same kind of organism. And the last one. Does anybody remember what the last one is? Anybody wrote it down? Just checking to see if you guys are still with me.
Adapt and evolve.
Very good. It is going to adapt and evolve. Adapt and evolve. Now, it's important to know that evolve means the population evolves. The population evolves, not the individual. Okay? Like individual things, like people cannot evolve themselves. Okay? It's the entire population that is going to evolve. So an example, just very briefly, okay, is that there are these little organisms, okay, in the population, and some of them have furs, okay, and some of them don't, okay, because of genetic variation. Just like people look different, right, in the human population. So these organisms, there are ones that have furs and then ones that do not, okay, and this is a genetic trait. And then let's say it gets really, really cold. It becomes minus, I don't know, 70 degrees or something. So that will kill off, this will kill off a lot of these ones that do not have furs, okay? Because, they get too cold. And then these ones with the furs will survive, and they in turns would have more offsprings than the ones without the fur, because the fur trait is heritable trait, something that you can pass on to the next generation. They will in turn produce more offsprings that will also have the fur. So over time, the population evolve to adapt to the cold environment. Okay. Now, if that cold environment becomes very hot now, for some reason becomes plus 70, all of a sudden, all these furry guys, they are not going to survive too well. Okay. And then the ones that are sort of like naked without the furs, they will then reproduce more, and then the population would evolve again. So evolution is not a fixed thing, right? It would keep on changing. The population would keep on changing so that they're better adapted to the environment. So the key takeaway for that is individuals do not evolve, population, they do. Any question before I move on? Feel free to interrupt, okay? Again, don't make this an interactive experience. Next one, we have a chart here, and there is something called induction and deduction, okay? So let me give you the definition of each, and then we will give you some example. So for induction means we will derive just means come up with, okay, derive general principles from specific observations. Okay, so this is basically, right, induction. I'm going to do here. Induction is like many small statements, and then you take all of this, and then you come up with a general statement. Okay, like a big general statement. Okay, so it's from many towards one, right? Kind of like this, like a triangle. Okay, going this way. Many general statements, and then you generalization, and many general statements, and then you try to come up with one general statement. Okay, so here is an example. Okay, here's an example. Where's my purple color? Right here. So I can say, An ant is made up of cells. Okay, I can say a flower is made up of cells. And then I can say a bird is made up of cells. A fish is made up of cells. So we make so many observations. A mushroom is made up of cells, an amoeba is made up of cells, a bacteria is made up of cells. And then we generalize and we say, right, we take all these things, right, take all these statements and we say, all living things are made up of cells. Does that make sense? Okay, induction reasoning. Okay, you take generalized statements and then you, or like many individual statements, and then you come up with one encompassing statement. Okay. Deduction is the other way around. Okay, so this one, the reasoning flows from general to specific. Okay. If you cannot read my writing, just let me know. Okay, it's not easy to write on the computer. Okay, so the reasoning goes from specific, general to specific. Okay, so if I do my triangle again, right, so I'm going to take one general statement and then I will apply it to various specific case. An example is that, okay, so we know from before, we said all living things are made up of cell. Okay, so that's general statement. And then let's say I discover a new organism, okay, our little furry friend from before. Okay, so now I'm gonna say this furry thing, right, since we'll just call it furry. Furry is a living thing. It is made up of cells. Does it make sense? The difference between induction and deduction, right? Induction, many things, and then you make up one general statement. Deduction, you start with a general statement, and then you apply it to many scenarios. Okay, it might not be just a furry thing. It might be you discover like another organism, and then you're like, since that is living, it is also going to be made up of cells. Any question? Okay, give me a second here. I'm gonna pull up the tutorial questions. I had that before my computer crash.
Sorry to interrupt, sir.
Yes.
Can you give an example of like a more complex deduction?
Yep, I am going to do that right now. So here is a bunch of questions, a bunch of scenarios, and you guys try it out first and tell me which one is deductive reasoning. I'm gonna launch the poll for you. Okay. Try that out. This would be an example of a quiz question. If you're not able to see the poll, you can just answer the question on your worksheet, okay? Okay, only half of you have answered. I'm going to wait for another few seconds before I close the poll. Okay. Now, I do encourage you guys to participate in these polls. It gives me an idea of, you know, where you guys are and if you're understanding the lesson. So here, let's take a look. Okay? We have kind of like a three-way split between A, B, and C. So let's take a look at this question. Okay. Deductive reasoning, right? So what is deductive? General statement, and then we apply to specific cases. Right? So the first one, all viruses requires a host to replicate. So that right here, that is a general statement. General statement. Right? It's like saying all living things are made up of cells. That's a general statement. And then it says COVID-19 is a virus. It must require a host to replicate. Right? So based on what we said in the study guide, we said the example I gave was all living things are made up of cells, and then the furry thing is a living thing. It must made up of cells. So this is the same kind of logic, right? So this would be the correct answer. But let's take a look at the other ones and see why they are not deductive reasoning. COVID-19, Ebola, influenza are viruses that require a host to replicate. Therefore, all virus requires a host to replicate. Right? So this is going from specific to general, right? You have many different examples. It's like the fish is made up of cells, the cat is made up of cells, flower is made up of cells. So therefore, all living things is made up of cells. So this is the wrong order. This is actually an example of inductive reasoning. Okay, so for that reason, B is not correct. Next one. All virus requires a host to replicate. COVID-19 requires a host to replicate. Therefore, COVID-19 is a virus. At first glance, this sounds a lot like deductive reasoning. But if you break it down and think about it using another analogy, then you will see that the logic is flawed here. So let me replace the word virus with birds. Okay, so birds. So I'll say all birds, instead of require a host to replicate, I'll say all birds have wings. Okay, all birds have wings. And then here it says COVID-19 requires a host to replicate. So instead of COVID-19, I'll say a bee has wings. Okay, this is basically what this statement is saying, right? So all birds have wings, bees have wings. And if I follow this logic, then what's my last statement? Therefore, a bee is what? A bee is. Do you guys understand what I'm saying? A bee is a bird, right? You see the logic here? Right? That's basically what this statement is saying, right? The logic is reversed. Okay, if this was proper deductive reasoning, it should be COVID-19 is a virus, therefore it requires a host to replicate. Okay, you can see the first one and the third one, they look very similar, but only one of them has the correct flow of logic. Okay, so I'm going to pause for a moment and see if anybody has any question about that. Good. And no one chose D, and D is not deductive reasoning. Okay, so because this says COVID-19 is a virus and it requires a host to replicate. Ebola is also a virus, therefore it also requires a host to replicate. There is no general statement going to specific. This is just one specific example and then another specific example, and there is no connection really between the two. All right. So someone asked for like a more complicated examples. I hope this answers the question. The student who asked earlier, is that okay? Or you need more examples?
Nope, that was good. Thank you.
All right, thank you. Moving on. List the steps in the scientific process. So we use the scientific process to investigate, right? Something that we observe in nature. And it's kind of like the standard process that we all use in the scientific community. So we start with observation, okay? And then question. Right? So you question what you are seeing, okay? And when we make observations, there are two types of observations we can make, right? Okay, there is the qualitative, and then there is the quantitative, right? Can someone tell me the difference between qualitative and quantitative? Perhaps give me some examples if you could. Anybody? Okay, so qualitative, it's like starts from the word quality, right? So when we talk about the quality of something, we use our senses to feel it. You're like, oh, this feels rough, this feels smooth, right? So those are qualitative. We use our five senses, the color of something, the odor, the smell of something, right? All these are qualitative. It doesn't involve measurements. It doesn't involve using numbers. Quantitative would involve using measurements. So these would be things like, size, mass, volume, speed, all those stuff will be quantitative, okay? As opposed to using our senses to make the observation. So you see a bunch of observations, you make a bunch of observations just in the natural world, and then you ask questions about it. And these are questions that you don't know the answers to, but you can make a hypothesis, which is a tentative answer for the question that you proposed, and then you will make a prediction. Okay, predict the results, and then you will do an experiment to test your prediction, test your hypothesis to see if it's correct, and then you will make a conclusion. Okay, and sometimes after the conclusion, you might reject the hypothesis or you might accept the hypothesis. And if you reject it, you're going to have to go back and make a new hypothesis and repeat the process. Okay, so those are the steps in the scientific process. If you need more information, you should watch the recording again. So in order for a hypothesis to be valid, it needs to be testable as well as falsifiable. Testable means you should be able to conduct an experiment to test it out. And falsifiable means it could potentially be wrong, okay? So let's start with an example of something that is non-testable. Okay, so non-testable. Well, anybody want to give me an example? You guys have been quite quiet. Anybody want to give me an example of something that is not testable? You can type it in the box if you want, or you can unmute. Okay, that's fine. I will do it myself. So an example of a non-testable hypothesis would be something like action movies are better than horror movie. Okay, so that is not a testable hypothesis because this is just an opinion. Okay, opinion cannot be tested. Okay, there's no right or wrong. Okay, you can't really say one or the other. It's not testable. Give an example of a non-falsifiable hypothesis. Okay, so if you work hard enough, then you will get A+ in biology. And this sounds like something my mother would say to me when I was a kid, right? If you work hard enough, you're gonna do well at school. Well, this is not a falsifiable statement. It's not a falsifiable hypothesis. Because if you end up not getting A+ in biology, the person can always just say, well, that's because you didn't work hard enough, right? Okay, so what's hard enough, right? Like this statement will never be false, because you can always argue the person hasn't worked hard enough yet if they don't get A+. Okay, so that's a not falsifiable hypothesis, and it's not a valid hypothesis. Okay. Moving on, if you guys don't have any question. Give 2 examples of primary sources and 2 examples of secondary sources. So primary sources are peer review journals, right? Okay, so when people submit their research, they are going to be looked at by other experts in the field, and then they make recommendations. And then if you meet the standard, right, of all these peers, then you would be able to publish it. Okay, so these are supposedly credible, more credible compared to something you read on a blog. Okay, another example of a primary source is a journalist reporting live from some kind of scene. Okay, so there is I remember seeing the tornado, right, that happened in Barrie. There were people reporting live, so that would be an example of a primary source. Okay, they are the first person there, and they're reporting directly from the scene. That is a primary source. Secondary sources are things that are not peer reviewed, or if they are extracted from a primary source. So for example, a textbook. Okay, so text material from maybe peer reviewed journals, and they summarize it and make it into easily understand material for students to read, for people to read. Okay, and then there are also blogs. So not all secondary resources are as reliable as primary sources, right? Particularly things that are not, checked. Textbooks are usually okay because they are, publishers gonna, check it before it's being published. Okay, but blogs, everybody can write a blog. I can write a blog, right? And it's not the most reliable things. And with all that's going on right now with the pandemic, right? People are worried about information that people post online, right? In blogs, in Facebooks, and so on and so forth about false information about vaccines, for example, and that is actually slowing down the effort to fight the pandemic. So secondary sources are not always reliable. Okay, number 6. I believe I did this in the lecture recording, so I'm not going to go through it again. You can watch the recordings to fill this in. But I will explain briefly the difference between independent and dependent variable. Okay, so the independent variable is the one that you have control over. Okay, this is the one that you deliberately, or we'll say you change on purpose. Okay, so you, in an experiment, you can only change one thing at a time, right? And the thing that you're changing on purpose, that is the independent variable. The dependent variable, okay, this is the one that you will be measuring or observing. to see whether it is affected by the independent variable. Sorry, it's very crowded here. Independent variable. Okay. So by changing the independent variable deliberately, you want to measure and observe and see if the dependent variable is affected. Sometimes it is not affected. There is no effect. Sometimes it might cause it to increase, to decrease, or whatever the case. Okay, so in an experiment, you have to decide what's the dependent variable and what's the independent variable. Okay. Now, perhaps I'll do one example. Students watched a cartoon either alone or with others, then rate how funny they found the cartoon to be. Okay, so the independent variable here, where the one that is being changed, would be watching alone or with others. Okay, so either with others or with yourself. And then the dependent variable, the one that you'll be observing or rating, would be how funny it was. Now, this is a little bit subjective, depending on the person, right? So you probably have to come up with some kind of scale, right? On scale of 1 to 10, how funny do you think this is? Okay, rather than just saying, you know, was it funny? Okay. Any question? Now, usually we're gonna have a break, you know, an hour in, but because today we spend almost half an hour doing the orientation, and then I had a little glitch at the beginning that took up some time. We will skip the break today, and we will cover as much as we could. All right, so all these things are in the videos. Like I said, you can look at them later. Now, let's talk about Rady and Pasteur's experiment on the theory of spontaneous generation. So what is the theory of spontaneous generation? Well, spontaneous generation states that living things came from non-living things. Okay, so it's all based on the observation that if you leave a piece of meat outside on a hot summer day, come back a couple days later, it's going to be creeping with maggots. So people say, oh, the maggots must have came from the piece of meat. Piece of meat is non-living. I mean, it was once living, but then it's like dead meat now, right? So then the meat, non-living, give rise to these maggots, the living organism. So that is an example of spontaneous generation. Okay? We obviously know this is not true, but at the time, people thought that was true. Okay, and you know, sometimes fish and frogs, they would come raining down from the sky. It's actually something that happened and still happens today sometimes. And people are like, oh, fish and frogs, they came from the sky, sky being non-living and the fish and frogs being living. The scientific explanation for that is because like there is some kind of you know, tornado or hurricane that sweep up these things from nearby ocean, and then they send them flying inland. That's the reason for it, but people didn't know that. So based on these observations, they're like, okay, let's come up with a theory of where living things come from, and they call it spontaneous generation. So Rady does not believe in spontaneous generation, and he wants to conduct an experiment to, you know, to check his beliefs. Okay, so blowfly experiment, living things arise from lifeless matter. Okay, I will check out what that means, and I will comment on that after. Thank you for sharing that. So Rady, Francesco Rady, that's the name of the person. Observe that. Observe that. Flies and maggots. Can be seen around pieces of meat that were left outside. Okay. So now he's going to do an experiment, and he has 3 groups. Okay. So the first group, he has a jar. Okay, so this is with a piece of meat inside. This is the meat. So meat in open jar. Open jar. Okay, that's the first group. And then the second group is same jar, same type of meat, but this time he's going to put a cover, a mesh cover on top. Okay, so meat in a jar with mesh cover, okay, with some kind of netting. And then group number 3, we are going to have a jar with a meat, but this time it is completely covered. Okay, completely covered. No openings. Okay, so meat in a jar with, we'll say in a sealed jar. Okay. So in this setup, what would you say the independent variable is? Independent variable. Anybody? What is the independent variable in this setup? What is the thing that he deliberately changed?
Where he would place the meat?
The lid?
Yeah, the lid would be well. He placed the meat in the jar, right? But then it's the type of lid, type of covering, that would be the independent variable. So good. So type of jar. Okay, and then the dependent variable would be the presence of flies and maggots. Okay, so he leaves these jars outside in the scorching sun, and then a couple of days later he comes back and he observed whether there are maggots or not. Okay, so what were the results of Rady's experiment? Okay, so we will have 3 groups. So group one, right? As you expected, flies plus maggots all over the place. Okay, it's one of the things I'm actually really scared of. Like sometimes, like once or twice every summer, like my garbage can is in my garage, but then once or twice every year in the summer, like they would be exploding with maggots when I take the garbage out, and then I would see these maggots. Like I'm so scared of them. My wife is not for some reason. So, you know, every time there's a maggot outbreak, then she'd be like, "Oh, I got this. You go back inside." I'm glad I have a wife like that. So she will clean it all up. Number 2, okay, number 2, this one has the netting, okay? So flies and maggots on netting only. Not going to be on the meat, right? So this one, you have the flies and the maggots right here. Like I'll use these blue things as maggots, and then you got the flies all over the place. And then here you have the flies, and then you have some maggots here, okay? but not on the meats. And then number 3, as you would expect, nothing, right? No flies or maggots. So based on this, right, by changing one variable, the type of jars that you have, he was able to show that the maggots do not come from the meat. Because if the maggots did come from the meat, you would have seen maggots and flies forming from this piece of meat that is sealed from the outside world, right? So based on this, he concluded that meat does not produce flies or maggots. Like you look at this and you're like, duh, of course not. This is so obvious, right? Okay, but remember, this is like, centuries ago. So at the time, right, people weren't really sure, and by doing these experiments, you are able to, figure out whether something is true or not. Okay, I'm sure the kind of stuff that we're doing right now, when people hundreds of years look back and they're like, obviously that was true, right? Why did you have to do an experiment to do it? And that's kind of like the point of all these. Okay, then there is Pasteur, okay, Louis Pasteur. So if you buy milk or cheese or whatever, or honey, sometimes it will say it is pasteurized honey, right? As opposed to like the raw honey. Milk are almost always, well, they are always pasteurized. You shouldn't drink non-pasteurized milk. So it's the same guy who came up with the process, the pasteurization. So this person, he also did an experiment. And what prompted him to do the experiment? So after Rady did the maggot fly experiment, not everybody accepted his findings. Okay, so many people, I assume it's people in the scientific community or like people in general, many people still believe in spontaneous generation, even though it does not apply to larger organisms. It still holds true for microorganism. Okay, so people are like, fine. We believe the flies and maggots don't come from the meat, but the microorganisms can still be forming from nothing. Okay, so like, if I have a cup of tea here, oh, you cannot see because I'm using a virtual background, but if I have a cup of tea here, I'm actually holding a cup of tea. Over time, microorganisms will form spontaneously from the tea. Okay. So what is Pasteur hypothesis? Well, so he says that if spontaneous generation is correct, then microorganism will arise from sterile broth. So broth is like, some soup, right? Chicken soup, beef soup, whatever. Sterile means it is like you heat it up, right? Heat up to kill everything. Okay, so like if you killed up everything with heat and then you put the broth away, if spontaneous generation was true, then you should see microorganism, a couple days later or whatever, right? So that's his hypothesis. So what is his experiment? Okay, so in his experiment, he has two set up. Okay, he has a flask with a sterile broth here, and then he has another flask, but this one has a swan neck. Okay, so what is the control group, and then what is the experimental group? Typically speaking, the control group is the group, right, that has no modifications to it. In this case, it would be this one. So control group. No special neck, if you would. And this is the experimental group. Experimental group. Okay, and it has like a swan neck tube. Okay, swan neck top, if you would. Right? So again, in an experiment, the control group is the group that is not going to be receiving any modification. Okay, so if you think about testing a vaccine, the control group will be the group that is not receiving a vaccine, and then the experimental group will be the one that is receiving it. Okay, there's also the placebo, which we'll get into in a bit. So what was the independent variable for Pasteur's experiment? What do you guys think? Anybody? The independent variable, the one that he deliberately changed in this case. Well, it's very similar to the previous one, right? It is the type of flask and lid used. Okay, so no lid, swan neck lid. Yeah, exactly, right? People are answering in the chat. Very good. How did Pasteur ensure the initial condition of all the flasks were identical? Well, he the broth was heated first to kill off all existing microorganisms. Okay, and then you put it in. So if you see new microorganisms in a few days, you know they are new. They came from they came from somewhere. Okay, because you killed up everything already. Okay, so that is how he ensured the initial conditions were identical. What were the results of Pasteur experiment? Well, as you would expect, microorganisms were found in the control, but not the experimental group. Okay. So as we now know, right, the microorganism can still fall in if it's open top, but if it's swan neck, right, then they will be able to get in here, but you know they don't really climb, right? So they cannot go up here, and the broth will remain sterile. Okay. So the conclusion is that microorganisms do not arise from sterile broth. Okay. Are we okay? Everybody still with me? The thing about online learning is I cannot see people's faces, so it's hard for me to gauge, you know, whether you guys are understanding. In a classroom, I can see, you know, if people are confused or not. But yeah, give me a thumbs up. Thank you. If you are okay, okay, you can click on the emotion or something, right? There's something, yeah, reaction. That's what it's called, right? Click on reaction, then you can. I will see like a little thumbs up. Oh, thank you. Oh, wonderful. I can see that you guys are still there. That makes me happy. Good. Okay, moving on. I hope this pace is okay, right? Sometimes I talk a little bit too fast. And if you want me to slow down, just let me know. So now we're going to do this one, long answer question. I will copy it to a new page to have more space. There we go. You are the lead scientist. Determine if taking multivitamin supplement reduces the risk of a cold infection. All right. So let's talk about how to set this up. First one, write a hypothesis for your experiment. Okay, so remember, a hypothesis has to be testable and potentially wrong. So I'm gonna write taking. There are really only two possible hypothesis for this case. Okay, so taking multivitamin, vitamin will reduce the risk of a cold infection. Okay, so either this is your hypothesis, or you will say it will not reduce the risk of a cold infection. Okay, either it will or it will not. Okay, like in the unlikely event, if you try to be creative, you'd be like, taking multivitamin will increase the risk of a cold infection. Okay, but logically speaking, right, this will be my hypothesis. Okay, next one. What is the independent variable and dependent variable? Okay, so independent variable, okay, would be whether the person is taking or not taking the supplement. Okay, so taking or not taking supplement. Okay, so the control group would be not taking, and then the experimental group would be taking the vitamins. And then the dependent variable will be catching a cold or not. Okay. So next one, what groups should be involved in your experiment? We talked about that already. You should have the control group, which is not taking it, and then you should have the experimental group, which is taking it. So next one, what factors must be kept constant between various groups? So give me 1 second. I had something opened earlier for you guys to try, but then when I restart, it disappear. So let me just pull that up quickly. It will only take half a minute. Okay, so in the chat box, I have posted a link. Why don't you guys go and click on that link, and you can tell me 3 factors that must be kept constant between the different groups here. Okay. Do you guys understand what you need to do? Okay. So you click on the link in the chat box, and then 3 standardized variables between the various groups. Does the link work for you guys? I am not seeing any response so far. All right, take your time.
What are we supposed to write?
So after you click on the link, there would be, hey, let me show you what it looks like. After you click on the link, there will be 3 boxes. List 3 standardized variables. So we have three. We have the experimental group, we have the control group, and we can throw in a placebo group, right? Okay. So we have people who are not taking the vitamins. We have people who are taking the vitamins, and then we have people who are taking like a sugar pill or something. In between these groups, what needs to be kept constant in order for this experiment to work out? Okay, so these are called standardized variables. Okay. Okay, I got one response, waiting for the others. Okay, good. So I just want to ask, right? Like I only see a few people answering. Is it because some of you don't understand the concept of standardized variable, or is it because there's trouble accessing the site? What's the situation? Okay, so let me explain again. If you're able to do it, great, please do it. If not, that's okay as well. So in this question, we are trying to determine if taking vitamins will reduce the risk of catching a cold. That's what we're trying to test, right? Okay. So how do you go about doing that? Well, we're gonna have 3 groups. Okay, we're gonna have 3 groups. Group 1, group 2, group 3. Okay, so we have the control group, we have the placebo, and then we have the experimental group. Okay, so each group is gonna have a bunch of people in it. Okay, a bunch of people. People, like this. Okay, many people. So what is the control group? Control group will have no vitamin. Okay, they're not going to be taking anything. Placebo group, you will give them a sugar pill. Okay, sugar pill. So you will tell them this is vitamin, but then actually it's not. Okay, so that will account for things like psychological effect or bias. Okay, and experimental group would be actual vitamins. Okay, my question is, what are the standardized variables for all 3 groups? So we must try to keep everything else the same, right? For example, in the meat experiment, we would want to use the same size jar, same type of material. You want the meat to be from the same day, right? Same size of meat, same type of meat. You want to keep everything the same except for the thing that you are evaluating. Okay, so it's much easier to, keep things constant when we're dealing with objects. It's a lot harder when we are dealing with people, because you're not going to have identical people in each group, right? But we try the best we could to keep variable standardized between these groups. Okay, so let's take a look at what some of the things people said are. Okay, so okay, so like type of brand of vitamins taken. So yeah, that would be important for the people who are taking the vitamins. Okay, however, when we talk about standardized variables, we are not just describing a particular group. We are describing all of the groups involved. Okay, so age group is good. You don't want all the old people to be in the control group and then all these, young, healthy people, younger people in one group, because then there is a bias, right? You want the age group to be very similar. Okay, it's like same as testing the vaccine, right? The COVID vaccines, right? That's why A12 and below is not approved yet, because when they test, they have to test it in like older people first, and then they run another test for like people in their 50s, and that's why it's being rolled out, by age group, right? So similarly, if we were to test this vitamin, we want each group, the control, the placebo, and the experimental group, to have people around the same age group. Okay, you want them to be having similar diet. Okay, you don't want the one group to be, eating takeout all the times, and then the other, cooking their own meals or whatever. You want them to have similar diets. Okay, maybe you provide meals for them, something like that. And the environment that they're in would be important. Okay, you want people to be exposed to similar environments. Okay, you don't want to put all the health care workers in one group and all the office workers in the other group, because the environment that they're exposed to is going to be quite different, right? So by nature, the healthcare workers might be more susceptible to catching a cold compared to the office workers, right? So you want to have a good mix in there. Okay, let's see any other good things here. Yep, health situation is also important, right? You want them to be relatively healthy in all groups. Be honest, yeah, that one is a little bit hard to gauge. But yeah, you don't want people. But if they're not going to be honest, they are not going to tell you that they're not being honest, right? So it's hard to adjust for that one. But I agree. A lot of these things is based on self-reporting, right? Did you get a call? And then they're like, no, I didn't. And then you just kind of have to take their word for it. Okay. Yeah, no drugs, no drinking. You don't want them to be engaging in activities that would potentially alter the results, right? So these are things that would be applying to all of the groups. Okay. Does everybody understand what standardized variables are now, or do you need more clarification? Okay, it's important you understand because I'm going to be asking you on the quiz. Okay, so there is a private message question. Would age and health be considered a standardized variable? Yes, they would. What about the background of the person? Yeah, exactly, right? Like background, as in like, I'm not sure what you mean by background, but like, for example, like ethnicity, right? Okay, that could be a background. A socio-economical background, that could be another thing, right? So, many of these things should be considered when you are dealing with people, okay? Because people by nature are not the same. Okay. All right, and if you still don't understand, again, you can email me. We can even set up like one-on-one help if you need to. Okay, like my job is just to help you do good, right? And don't be hesitate to reach out and contact me. Okay, people haven't been contacting me in ever since we switched online, and that's just not good, because you're making my job too easy. Right? You want me to work for my salary. Okay, so make sure you contact me and keep me busy and ask me questions if you have any. Okay, I will definitely help you if you need. Okay, moving on. Well, what results would support your hypothesis? Well, if we choose to say that the vitamin will reduce the risk of cold, then what we should expect to see is the group that is taking the vitamin would have substantially lower number of cold infection. Okay, so if we see a lot of people in the experimental group not catching the cold, then we know that taking supplements will help, okay? On the other hand, if we see similar numbers in all groups, okay, let's say, 70% of people in each group still catch the cold, then we know, taking supplements is not going to help, okay? So yeah, that answers E and F. Now, before we move on to the next one, last call. Anything else to clarify? No, we good? Yeah. Great. I will take your word for it. Okay. Let's see if there is a poll that we could do. Okay, let's try this one. Let's try this one. I will relaunch the poll. Go ahead. Okay, I'll be right back. This one takes a bit of time. Okay, so it has almost been 2 minutes. Got some response, so I'll end the poll because we still have a lot to do. Share results. Okay, so here we have 1 ml of an experimental drug diluted in saline solution. Saline solution just means salt, right? Okay, and is injected in 20 pregnant mice to determine possible side effects. Which of the following is a suitable control for the experiment? Okay, so remember, control and an experimental group, at the two groups, there should only be one difference between the two groups. Okay, so here, 20 male mice injected with 1 ml of saline. Okay, so this is clearly not the answer because we're trying to determine possible side effects for pregnant mice. Male mice not going to get pregnant, so anytime you see the male mice, you're going to get rid of them. Okay, non-pregnant mice also will not be suitable for this experiment. Okay, because we again, we're trying to assess the effect on pregnant mice. Okay, so now we only have 2. Okay, so 20 pregnant mice injected with 2 ml of drug, 20 pregnant mice injected with 1 ml of saline. Okay, so the control group versus the experimental group. Right, experimental group is the one receiving the drug. For the control group, they should not be receiving the drug. So this is not the answer, and they should only be receiving the saline. Okay, because the saline is the thing that is being diluted in, right? So it's again like the vaccine. I keep on bringing the vaccine because it's the relevant thing that goes on right now, right? So if you test the vaccine, say the mRNA vaccine, the control group versus the experimental group would be the entire vaccine is the same, except in the control group, it will be missing, say, the mRNA, Okay, so that would be control versus experimental. The correct answer for this is E. Okay. This question will be on your quiz, so make sure you understand it. Okay? Maybe with subtle difference. Okay, moving on. Where were we? Next one. A farmer wants to determine if feeding cows with high protein diet would increase milk yield. So hypothesis, what is the hypothesis here? Again, the hypothesis should be the easy part, right? Okay. You either can say feeding the cows with high protein diet will increase milk yield, or you can say it will not. Okay. Either of them is okay, right? A hypothesis is just a guess, right? An educated guess. Some people say a tentative answer. So that's the hypothesis. It will or it will not. Describe how the experiment should be set up in this hypothesis, okay? So we're gonna have cows that are fed with high protein diets, okay? And then we're gonna have cows with regular diet or low protein diet. And then we are going to measure the milk yield. So in this case, the independent variable is going to be protein level in diet, okay? And then the dependent variable will be milk yield, okay? Should we include a placebo in this experiment? What do you all think? Placebo, yes or no? If you think yes, choose A. If you think no need for a placebo, please choose B. That looks more like a pig than cow, sorry. Never mind, I will not draw the cow. So we have the groups with the high protein diet, low protein diet. Control experiment, should we put a placebo group? Okay, so we are kind of split here, half-half, right? You know, almost half of you say we need a placebo and then the other half say we don't. So let's hear some explanation, okay, for people who say you do not need a placebo. Anybody want to justify your reason? Anybody want to share why you chose no placebo is needed? I want to hear the reason.
So my reasoning was just that it's pretty straightforward with what he's asking for. It's either high protein diet will increase it or not. So if there's only two groups I think that we would need. If we add a placebo to it, there shouldn't be a result. It would just be regular, that like what I guess the cows are getting right now.
Okay. Yeah, thank you, Michelle. Anyone else want to chime in of whether we need? You can either explain why you think we need or we don't need a placebo. Okay, so that was good. For Michelle.
I would say no to the placebo because the cows aren't going to be biased on the outcome.
Yes, that's the very good answer, Rita. There, that is the exact reason. Okay, like unlike the humans would have bias, right? Like, you know, my daughter, she would, you know, fall on the ground and then, you know, scrape the knee a little bit. It's not even bleeding, and she'd be like crying and screaming, it's hurting, blah 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 blah, right? And then I put a Band-Aid on. The Band-Aid does absolutely nothing other than psychological comfort, right? As soon as I put the Band-Aid on, and she'd be like, I feel better now. Okay, so that's like psychological effects, right? So sometimes when we do experiments with people, you know, you give them a pill, and then, you know, because they think they're taking something, they will tell you, oh, I feel better, right? So to account for that kind of psychological effect, that's why we need a placebo. Okay? That's why we need a placebo. Cow's not gonna have that problem, okay? The cow's not even gonna know you give them high protein or low protein. They're not gonna be like, oh, I'm gonna hold out on the milk production for now, okay? Because I know you are experimenting with me. So cows don't have that problem. For that reason, we do not need a placebo for this particular experiment. Okay, so let's try that one more time. Okay, I want to see you guys do this one more time for me, please. Okay. So go to the Mentimeter thing, and I want you to type in the standardized variables for me one more time. Okay. Because I know last one people were not entirely sure. So I'm going to ask you to do that one more time for me, please. Okay.
Sorry, sir, can you repeat the question?
Yes. So in the chat box of the Zoom, right? If you move over to the ribbon, there is a chat, like a little speech bubble. If you click on that, you will see that I've typed in a link. Okay. So click on that link, and it will take you to the to this page again. Yeah. Okay. So I want you to type in 3 standardized variable that I should have for this cow experiment. Okay. So we have 2 groups, right? Cows with high protein versus cow with regular protein. Okay. Everything else should be the same. Okay. So I want to know 3 of those standardized variables. Okay. For the cows. Okay, good. I have some answers here. Okay, I think I will pause there. Let me see. Okay, so there we go. Okay, we only have 5 people answering. Maybe the rest of you are still trying to type in the answer. That's fine. So same outdoor activities. I love that. Okay, you don't want to lock up the cows who's not having high protein, and then the other ones get to go out and free range. You want them to have the same outside exposure. The type of proteins you feed them is also important. You want to have the same feed for both groups. It's just the amount that you're getting varies. Okay. Same type of protein. That's good. Behavior of the cow. Not quite sure what that means. You can't really control how they behave, but I think maybe you mean like how you are treating the cows, maybe, right? Like you want to give them the same condition, the same environment. Age of cow is important. Very good. Health of cow, body weight, very good. Same milk being given. We are trying to measure the milk though. Okay, we're not giving them the milks, right? We're trying to measure the milk yield as a result of a change in diet. Okay, the number of times you feed them, for example, very good. Now, I am very glad that nobody mentioned about gender. Okay, like the gender of the cow. When I put this question on like an actual test paper, when we were doing face-to-face, many people put gender, and that would be wrong because male cows don't make milk. Okay, so in this particular experiment, all the cows that you use would have to be female cows. So that's just one thing that people usually get wrong. Okay, now we do not have time to go through all of these. Do you guys have class like right away after my class here? Like after like at 10:30 or something like that? Can you guys just let me know? Do I have to do another poll? Okay, I'll do another poll. Relaunch. Okay. Immediately after my class at let's say 10:30. Do you guys have another class? Or like at 11 or whatever? Can I go over a little bit, in other words? Okay, some of you do. Okay, yeah, that's fine. Okay, so I'll try to end at 10:30, and if I go over a little bit, if you have to go, you can go. And then you can watch the recordings after. Okay. Again, I apologize for the interruption at the beginning because of the computer glitch. Okay, describe the main principle of experimental design. Okay, so there are a couple of things here that you need to consider. Okay, so we need to have, for example, proper control group, control, placebo, and experimental group. Okay, you have to have a control group to compare your results to. Okay, you can't just give everybody the vaccines and then try out the results, because there's no baseline to compare it to, right? So that's why you have to have the control. Okay, and then there are, later on in the course we will learn about like positive control and negative control. Okay, but in general, you have to have control groups. You want to make sure that that there is a standardized variables. Okay, and we want to have randomized participants. Okay, you want to have a good mixed groups while keeping in mind the standardized variables. And then also you want to have a large sample size. Okay, you don't want to study that only have 10 people, right? You want to involve as many people as possible, because statistically speaking, the bigger the sample size, the more accurate your result is going to be. So, if you have been following the whole vaccine rollout news, right? You might remember hearing people keep on saying the efficacy of the Pfizer vaccine is like, 97% protective. And then for the AstraZeneca, it is like, 75%, right? You guys remember hearing that, right? And then they're saying, don't pay too much attention to that efficacy number, because that's based on a relatively small sample size compared to what's happening in the real world. Once those vaccines are applied to the real population, then all of a sudden the sample size becomes much bigger, and those percentage changes. Okay, so in other words, the more people you are looking at, the larger the sample size, the results are going to be more reflective of what is the true expected outcome. Okay. We talk about the importance of having a placebo. What is the difference between a blind experiment and a double-blind experiment? Okay, so in a blind experiment, the participants do not know what they are receiving. Okay, they don't know if they are receiving the placebo or if they're actually receiving a treatment. Okay, and that's important. Okay, because if they know that they're receiving a placebo, then that would not account for the psychological effect. Okay, because they know they're receiving for something that's not real, then they might think that it's not going to work. Okay, so they don't know, but the person who is administering the test, they would know. So in a blind experiment, only the participants will be shielded away from what they are receiving. All right. The gold standard of performing experiment is a double-blind experiment. So in a double-blind experiment, okay, both the researcher and participant do not know what they are giving or what they're receiving. Okay, sometimes the researcher themselves have bias. Okay, like if I develop a new drug and I work for this pharmaceutical company, I want it to work, right? I want my drug to work. So when I evaluate the symptoms, I might have subconscious bias when I interpret the results. And so in a double-blind experiment, I also don't know who's receiving what. Okay, so that information is kept, by some, I don't know, third party. Okay, and then we can correlate data later on. Okay, so double-blind is the gold standard. Does everybody understand blind versus double-blind? Perfect. Okay, now I am gonna skip this one for now and focus on number 2 because this one is quite confusing for many people. Okay, and this is explained in the lecture recording. So if you haven't already, you can watch that later on. So here I'll read it to you. A new drug called sleep is being tested for its effectiveness in reducing symptoms of jet lag. A double-blind experiment was conducted. The graph below shows the experimental data from the study. Based on the experimental results, what conclusion can be made about the effectiveness of sleep in reducing symptoms of jet lag? Okay, so let's do a quick poll. I know we don't have a lot of people here, but we will still do a poll. If you think the drug is effective, please choose A based on the results, okay? If you think it is not effective, please choose B. Okay, I will stop soon. 2 more people, waiting for 2 more people. No. Okay, yeah, I'll stop there. Okay, so more people think it's effective than people who think it's not effective. So let's take a look at how to answer this question. Okay, we have 3 groups here. We have the control group. Okay, so control group means not taking the drug, right? So no pills, no pills. Okay, and then the placebo is taking a sugar pill, or we can call it like a fake pill. Okay, we will tell them, hey, this is the new drug, the sleep drug. Okay, you take it. Okay, but then it's actually not. It's just like sugar maybe. Okay, and then this is the actual pill, right? The sleep, right? Sleep. Okay, so now does it work? Well, control group, people who didn't take any pills, 70% reported symptoms of jet lag. That is expected, right? Okay, they didn't take the pill, so nothing was helping them with the jet lag. You would expect a lot of people to have symptoms of jet lag. Okay, and then I want you to kind of ignore this for now and just focus on the last column here. So these are the people who are taking the pill. Okay, so they took the pill, and then only 40% or less, a little lower than 40%, only them reported jet lag symptoms. So compared to control group, it appears that the pill is helping, right? But we must also look at what happens to the placebo group. The placebo group, you give people the fake pill. You tell them it's the new drug. It will help you with the jet lag. They take it, and then you look at what happens. And you can see the placebo group also reported a reduction in jet lag symptoms. Okay, so what does this tell you? It tells you that the pill is actually not doing anything, because you could give people a fake pill to achieve the exact same result. So this shows you that the jet lag is more like a psychological effect. The pill is not helping because you could have given them a sugar pill and it would work just as well. Okay, it doesn't matter that this is lower than the control group. What matters is this perform in the same level as the placebo group. As such, it is not effective. So the treatment group and placebo group both reported lower percent of jet lag symptoms compared to control. Therefore, not effective. Okay. If the drug was effective, then the graph would look more like this. Okay. If the pill is effective, then the graph would look more like this. Okay. Let me see if I can do this. This one would be similar to the control group. You give them the fake pill and then you tell them it's the real drug and then they take it and then there's no effect. It should have no effect because it's a fake pill, right? You know what I mean? So if this was the case, then you would know it is effective. Are we okay with this? This is a little bit confusing. So if you have a question, please ask now. Okay, thank you, Michaela, for the comments. Okay, so sit on this a little bit, and if you find it not making sense later on, we could go through that again. Okay, so I know it's 10:30. If you gotta go. I'm just gonna quickly go through the other question as well as the fill in the blanks. It should take about 10 minutes or so. If you have to go, our next session is next Friday, right? On the Tuesday, you're supposed to do your own asynchronous learning for lecture number 2. I'll post an announcement after class, so you would know where to get the stuff. Okay, so the other question, the graph below. Okay, so it shows the effect of seeing lozenges in reducing cold symptoms duration. Identify additional resources and information that needed to improve the credibility of the results. Okay, so here is a graph that shows the placebo lozenges, and then we have the zinc lozenges here, and then the number of days until the cold symptoms are gone. So a couple of things that we would want to know about this experiment is, for example, who's paying for it? Who's paying for the study? Okay, is it the pharmaceutical company who is producing this? Is it funded by public health? Is there a financial conflict interest here? Okay, so we want to know that. We also want to know things like sample size, right? Sample size, right? It didn't say how many people were involved. Was it just like 4 people, 5 people, right? Even 100 people might not be enough, right? We also want to know things like how were cold symptoms reported? Okay, could it be confused with allergies? Is there a checklist that people use? How is it being evaluated? So all these additional informations would help us determine if something like this is credible or not. Okay, it's like those infomercials, right? They always like to throw graphs on the screen and say, it's scientifically proven, right? Like CoFX, right? I mentioned that in the lecture recording. It turns out to be not very credible scientific research. Okay, quickly fill in the blanks. Something is the ability to maintain a stable internal environment. That is homeostasis. Okay. part of scientific, part of discovery science, where scientists make generalization from many small observations. So that's like going from specifics to general, and that is inductive reasoning. Okay, induction rather. Okay, or inductive reasoning. The next one, that uses to make predictions about specific cases based on generalization. So start with a broad statement, and then we apply it to individual cases. That is deduction. A hypothesis is a tentative, testable explanation for an observation. And for the hypothesis to be valid, it should be testable and falsifiable. This variable is being measured or observed, so that is the dependent variable, like the milk yield from the cow. And then the one that you are changing or it's being manipulated, that is the independent variable, like the amount of proteins in the feed for the cow. A sham treatment, or like a fake treatment, is called a placebo. And we need to have a placebo to eliminate psychological effects and bias. A blank study is one where subjects are the participants, do not know if they're receiving a treatment or a placebo. We call that a blind study. And if the person who's doing the experiment also don't know what they're giving, then it is called a double blind. Something is a relationship between two variables, that's correlation. Primary source is a researcher's original report, whereas books and news reports, these are secondary resources. Okay, so that's it for this week. Thank you all so much for coming, and thank you for your attention. Again, feel free to reach out to me during the week if you have any questions, concerns, or need extra help. Otherwise, I'll see you next Friday at 8:30 again. Okay, have a fantastic weekend. See you next time.